You're afraid of something you've never experienced. Maybe you've always known it. Maybe you've been carrying it so long it feels like personality, like a quirk. Like just the way you are, the thing that makes you flinch when nobody else does. The image that hijacks your brain at 2 a.m., that specific, oddly precise, completely sourceless dread that lives in the basement of your chest and never fully leaves. You've explained it away, you've rationalized it, you've Googled it at midnight and convinced yourself it's fine, it's normal. Everybody has their things, right? You've explained it. But here's the part that nobody told you. It might not be yours. That fear, that specific ancient paralyzing terror that lives in your body like it was built into the architecture, may have been packed into your cells before you existed, before you were a thought, before you were even a possibility, wrapped in a chemical tag the size of a molecule, folded into your DNA like a note your great-grandmother slipped in your pocket before you entered the world. The note reads, "Watch out. I didn't survive this so you could walk into it." Welcome to the Phantom Navigator. I'm your host, and tonight we're going somewhere that is going to rearrange how you think about yourself. We are diving into the science of inherited nightmares, the molecule biology of the family curse. We're going to talk about why your bloodline might be haunted not by a ghost in a hallway, but by a ghost in your genome, in the actual chemical architecture of your cells. And then, because I refuse to leave you just wrecked in a ditch, we're going to talk about what happened, talk about what 2025 and 2026 have brought us in terms of actually doing something about it. We are covering epigenetics, ancestral trauma, mice who were born afraid of flowers, Holocaust survivors, Irish famines, Civil War prison camps, the 8% of your DNA that is prehistoric virus. Yes, you heard that right. We'll get there. A government conspiracy theory that should probably make you significantly more paranoid than you already are. And technology that can read the molecule address of a ghost that entered your bloodline a century ago. This is not a metaphor episode. We are being completely devastatingly literal. Buckle in. This one gets uncomfortable. And that discomfort you're already feeling, that's ancestral. You come by it honestly. Let's start with a question that has haunted humans since we first started keeping records of hauntings. Why does catastrophe echo? Why is it that the descendants of the enslaved carry anxiety profiles distinct from those whose ancestors were not enslaved, even generations removed from the original trauma? Why do studies of Irish American communities show epigenetic metabolic markers that trace back to the famine across people who have never set foot in Ireland and whose grandparents never talked about it? Why do the children and grandchildren of Holocaust survivors show neurological stress profiles that mirror their parents in ways that cannot be fully explained by shared cultural environment or conscious parenting? Hey, I don't think it's coming in. Thank you for that. Why do family curses work? Not metaphorically, not poetically, but practically. In the actual, observable, repeating patterns of anxiety, dysfunction, fear, and inexplicable physical illness that move through bloodlines like water through cracks in stone. For centuries, every culture on Earth had an answer. They called it by different names: miasma, blood pollution, ancestral sin, the family hex, the inherited wound. But the fundamental claim was the same. What happens what happens to you gets passed to your children, and their children, and their children's children, in ways that go beyond what you consciously teach them. Modern Western science spent a very long time mocking this idea, dismissed it as superstition, as a primitive pattern matching, as the comforting mythology of people who didn't understand genetics. And then, in the 20th century, a field called epigenetics began to take shape. And one by one, the mockery got very, very quiet. Because here's what epigenetics revealed: the ancestors were right. Not symbolically, chemically, biologically, molecularly right. Let me lay down the framework, because this is the foundation everything else builds on. You have heard of genetics. You know the basics: DNA, the double helix, the sequence of chemical letters, adenine, thymine, cytosine, guanine. That spells out the instructions for building and running a human being. That sequence is remarkably stable. A mutation that actually changes a letter in the sequence takes many generations to establish itself in population. Your genetic code is essentially the same as your parents', mostly the same as a chimpanzee's, if we're being humble about it. But genetics is not the whole story. Genetics is the script. And epigenetics, the field that is currently rewriting everything we thought we knew about inheritance. Epigenetics is the director. The director doesn't change the word on the page, but the director decides which lines get shouted and which gets whispered. Which scenes get cut, which characters get to speak at all. Epigenetics is the layer of biological instruction that sits on top of the genetic code, controlling which genes are active and which are silent without altering the underlying sequence. And unlike the DNA sequence, which is relatively fixed, epigenetics marks are dynamic. They respond to environment, to experience, to catastrophe. Here is the mechanism. Pay attention, because this is where the ghost enters the machine. When a human being experiences severe trauma, and we are talking about the kind of survival level terror that reads the nervous system, as this species may not continue, the brain triggers a massive stress response: cortisol, adrenaline, glucocorticoids, flooding this system in quantities the body was never designed to sustain long term. This hormonal cascade has downstream effects that should, by the old rules of genetics, be impossible. It reaches the DNA. Specifically, the hormone flood activates enzymes called DNA methyltransferases. And the enzymes do something extraordinarily specific. They attach tiny chemical tags Methyl groups a carbon atom bonded to 3 hydrogen atoms, CH3, to specific regions of the genome. 2 regions called CPG islands, which act as a control switch for gene expression. Think of it this way. Imagine your DNA as a massive electrical panel, thousands of switches, each controlling a different function. A methyl group is a piece of tape over a switch. Tape it down. The switch stays off. The gene is silenced. Tape it up. The switch stays on. The gene is locking into continuous activation. Now here is the critical question. Which switches are getting taped? That answer is what makes this terrifying. Among the genes most vulnerable to trauma-induced methylation are the genes that regulate the brain stress responses. Genes like FKBP5, the gene that controls how efficiently the body responds to and recovers from cortisol. Genes like NR3C1, the glucocorticoid receptor that tells the brain when it's safe to stand down. When these genes get taped, when the stress-regulating switches get jammed, either silenced so the alarms never turn off, or hyperactivated so the brain never stops anticipating threat, the entire nervous system recalibrates. It resets its baseline. And that reset baseline now defines what this person's body considers normal. Fine, one traumatized person. Awful, but contained. Except, and here is where Pandora's box opens, the trauma doesn't stay contained to the individual who lives through it. When a severely traumatized person reproduces, those methyl tags, those epigenetic switches that got jammed during the catastrophe, have a disturbing tendency to survive the journey to the next generation. Here is why that should be impossible, and why it isn't. At fertilization, the embryo undergoes what is called global demethylation, a factory reset. The biological assumption. The assumption that underpinned genetics for decades is that very new life begins clean. The epigenetics marks accumulated by the parents during their lifetimes gets wiped. The embryo inherits the DNA sequence, but not the experience encoded into it, except the reset is not complete. There are regions of the genome, imprinted regions, that are specifically exempt from demethylation. They are protected, preserved, and there is a second vehicle for smuggling information past the reset. Small non-coding RNAs, specifically microRNAs and tRNA-derived small RNAs, which live in the cytoplasm of the sperm cell itself, invisible to the demethylation machinery. These tiny RNA molecules act as a biological contraband. They ride inside the sperm, slip past the border checkpoint of fertilization, and arrive in the developing embryo, carrying their cargo of ancestral experience. The child is born with stress response genes reconfigured to match the conditions their parents survived. Not the conditions the child will live in, the conditions the parent survived. The child's alarm system is pre-calibrated for a danger that may no longer exist. The switches were taped before the child drew their first breath. The baseline was set before they had a single experience of their own. Your nervous system was configured for someone else's war. And the devastating poetry of it is this. It was done for you. It was an act of biological love. Your ancestor's body, in the midst of catastrophe, used every mechanism available to send a warning forward in time, to give you a head start, to make sure that if that danger ever came back around, your body would already know what to do. The tragedy isn't that the danger didn't come back, but the warning did. You are walking around in a world that is, relatively speaking, safe with a nervous system that has been preloaded with someone else's survival response to a world that was not. That is the architecture of bloodline curses, not mythology, chemistry. Now I need you to tell you about the most elegant horror story in modern neuroscience. Because everything I just described sounds theoretical until you see it demonstrated in a laboratory, clearly, repeatably, irrefutably. The year is roughly 2013. Brian Diaz and Carrie Ressler, researchers at Emory University, published a study that sounds like something a Victorian Gothic novelist would have written if they'd had a PhD in molecular biology. They took male mice, healthy, normal, no previous trauma, and they did the following. They exposed these mice in the sense of acetophenone, a chemical compound that smells like cherry blossoms or almonds, depending who you ask. Sweet, floral, completely harmless. And it simultaneously, they delivered a mild electric foot shocks. Classic Pavlovian conditioning. Smell the flower, receive the pain. The mice were fast learners. It didn't take long before the mere introduction of cherry blossom scent, no shock, no pain, nothing, was enough to send the mice into a state of visible, measurable panic, freezing, startle responses, the full physiological profile of a fear response. Fine, expected. That's basic conditioning. That's not the story. Here's the story. The mice had children. These offsprings were raised in completely controlled environments. No cherry blossom scent. No foot shocks. No contact with their traumatized fathers after birth. In many trials, artificial insemination was used to eliminate even that variable. No possible mechanism for social learning or observed behavior. The children had no way of knowing their fathers had experienced anything at all. And then the researchers introduced a sense of cherry blossoms, panic, freezing, a visceral, instinctive, unmistakable fear response to a flower they had never encountered, from a father they had never meaningfully interacted with. And then the grandchildren, same controlled environments, same absence of any direct exposure to the original trauma or conditioned fathers. Same response. A fear transmitted across 2 generations in the complete absence of any direct experience of the thing being feared. But here is the part that elevates this from impressive to genuinely disturbing. When researchers examined the brains of the descendant mice, the children and grandchildren, they found that the fear had not merely been passed down as a vague neurological sensitivity. It had been physically built into the anatomy. The descendant mice had grown larger nerve centers, specifically larger glomeruli in the olfactory bulb. The brain structure responsible for processing smell, and these enlarged structures were specifically dedicated to detecting acetophenone. The cherry blossom receptor infrastructure in their brains was physically bigger than the mice with no ancestral exposure. The fear restructured the anatomy of the brain before mice were born, before their first experience of anything. And when scientists examined the sperm DNA of the traumatized fathers, they found the molecule fingerprints, a targeted demethylation and unsilencing of a single specific gene, the OLFR151 gene, the exact gene that codes for the cherry blossom odor receptor. One trauma, one specific sensory input, one precise genetic address, one clear transmission, father to child to grandchild, via a mechanism so specific it reads less like biology and more like intentional engineering. The fear wasn't a vague psychological vibe passed through some mysterious mechanisms. It was a hyper-specialized, precisely targeted, physically manifested upgrade to the sensory architecture built into the next generation's nose and brain to ensure they would detect this specific threat faster than any unexposed organism could. It was, in the most literal biological sense, a warning written into the flesh of children who never heard it spoken aloud. Now, let's sit with the dark comedy angle here for a second, because I think we need to acknowledge it. These mice, the grandchildren especially, were just living their lives, just being mice, doing mouse things. And one day, someone introduced the smell of cherry blossoms into their environment, and they had a complete meltdown about a flower. From the outside, that looks irrational. That looks like a mouse with anxiety problems. From the inside, from the molecular biology perspective, that is a mouse whose grandfather encoded a survival warning into its genome with such precision that the specific scent of a specific flower triggered a precisely calibrated alarm system before the mouse had a single memory of anything. The irrational fear is the most rational possible response to information the mouse's brain received before birth. Sound familiar? The mice are powerful. The mice are convincing. The mice are mice. They can be shocked and controlled and studied in ways that would be, to put it gently, frowned upon if we tried them with human participants. How do we trace this in humans? Enter Dr. Rachel Yehuda, Mount Sinai Hospital, New York, a researcher who has spent decades doing what might be the most important and most heartbreaking work in modern psychiatry, studying the biological inheritance of the Holocaust. Let me set the stage for you. The Holocaust is, from a research perspective, one of the most devastating natural experiments in modern history. Not because any sane person would design it. It was evil. Categorically, incomprehensibly evil. But because it produced a large, relatively well-documented population of trauma survivors with identifiable descendants across multiple generations, in a country where medical record keeping was meticulous, Yehuda and her team focus on a specific gene, FKBP5, the gene that regulates how efficiently the body responds to and recovers from cortisol, the stress hormone. FKBP5 is essentially the volume knob on your stress response. When it's working properly, cortisol spikes in response to threats and subsides once the threat passes. When FKBP5 is dysregulated, either silenced when it shouldn't be or stuck in permanent activation, the cortisol response goes haywire. The alarm doesn't turn off correctly. The nervous system stays in crisis mode even when the crisis has passed. Yehuda's team took blood samples from Holocaust survivors. They found, unsurprisingly, distinct methylation changes on the FKBP5 gene. The survivors' stress response systems have been chemically altered by what they have experienced. The switches have been jammed. Expected, logical, devastating, but scientifically predictable. Then they took blood samples from the survivors' children, people who were born after the war, people who had not been in the camps, had not experienced the deportations, had not witnessed the violence, people who had grown up in, relatively speaking, safety. And they found mirrored methylation changes in the same gene. The same switches flipped in the same direction on the same gene in people who had not lived through the original events. The children's HPA axis, the hypothalamic pituitary adrenal axis, the body's central stress regulation system, was structurally preset to high alert, not as a choice, not as a learned behavior, as a biological inheritance. Their bodies were, at a cellular level, anticipating a war zone they had never entered. The researchers were careful, exhaustively, methodologically careful to account for the alternative explanation that the children showed elevated anxiety and PTSD vulnerability simply because they were raised by traumatized parents. Because the family environment, the stories told and not told, the hypervigilance modeled by their mothers and fathers shaped them psychologically. They couldn't fully separate those variables. That's the honest truth of the human epigenetic research. We can't do the controlled mouse study on humans, but the biological markers they found, the specific methylation patterns on the specific gene, showed up in the children even when controlled for what was observable in the environment. The biological signal was distinct from what could be accounted for by parenting alone. The ghost got into the genome, and then the genome got into the children. Now I want to go a step further, because Yehuda's work is extraordinary, but it's not alone. Research on the descendants of American slavery has shown elevated rates of cardiovascular disease, hypertension, and anxiety disorders in Black Americans that persists even when accounting for current social economy conditions and conscious stressors. Researchers are increasingly looking at whether multi-generational epigenetic inheritance plays a role alongside the very real and ongoing impact of structural racism. Research on Cambodian refugees and their children shows elevated PTSD vulnerability in the second generation. Research on descendants of famine survivors, which we are going to get very deep into the moment, shows metabolic alterations that match what you'd expect if the body were still running a scarcity protocol from an event that ended before the current generation was born. The pattern is not isolated. It is not one study, one population, one gene. It is a pattern that keeps emerging across different cultures, different catastrophes, different continents, different decades of research. What happened to them is in you, not as a memory, not as a story, as chemistry. And here is the thing I find most important about this, and I want you to hear this clearly because it changes the frame on a lot of suffering that gets pathologized and misunderstood. If your anxiety seems disproportionate to your current circumstances, if you feel a dread that doesn't match your life, a hypervigilance that fires in space that are objectively safe, a terror that has no traceable or origin in your own experience, you may not be malfunctioning. You may be receiving signal that was sent before you were born. You may be the recipient of a biological intelligence about a danger your family survived. The tragedy is not the signal. The tragedy is that nobody taught you how to read it. Here is something I find genuinely beautiful in a deeply unsettling way that I find most beautiful things. Before the microscope, before the sequencing machine, before the methyl group had a name or a chemical formula, before any of this science existed, human beings already knew. They didn't have the vocabulary, they didn't have the mechanism, they couldn't explain why the curse ran in the blood, only that it did. But across every culture on Earth, across every language, across every century of recorded history, the same fundamental observation kept surfacing. What breaks your ancestor breaks something in you. And what breaks you will echo in the ones who come after. The ancient Greeks gave us the House of Atreus, one of the most enduring horror stories ever committed to writing. A multi-generational saga of inherited doom that spans Tantalus, Pelops, Atreus, Agamemnon, Orestes, and beyond. Ending people, betrayal, cannibalism, madness, infanticide, revenge. Each generation pulled inexorably into the same patterns, the same triggers, the same violence, as if cursed by the gods themselves, condemned to repeat a catastrophe they personally did nothing to initiate. Aeschylus, Sophocles, Euripides, the greatest playwrights of ancient world all returned to this material again and again because it resonated, because their audiences knew this pattern, because they had seen in their own families, their own communities, their own bloodlines. They called it miasma, blood pollution, a stain on a family that traveled through generations, warping the descendants of the guilty party, even when those descendants were entirely innocent. We now called it transgenerational epigenetic inheritance. The Greeks got there first. They just described the effect rather than the mechanism. The Old Testament codified it, God visiting the iniquity of the fathers upon the children unto the third and fourth generation, which sounds like a divine punishment until you squint at it sideways and realize 4 generations is exactly the time frame that early epigenetic research identified as the window of transgenerational transmission. Not a coincidence, possibly the oldest documented attempt to warn a population about hereditary trauma using the only communication technology available, theology. Now let's get specific. Let's get historical. Let's put numbers on the table. Nazi occupation of Holland, a deliberate blockade of food supplies, an entire population reduced to consuming tulip bulbs and sawdust to survive. Winter, starvation, pregnant women surviving on fewer than 500 calories a day. Their children were born, seemed on the surface, fine. Perhaps small, perhaps fragile, but alive. These children grew up, had children of their own, lived their lives. And then the long-term studies started coming in. 60 years after the Dutch hunger winter, the descendants of those in utero famine survivors showed altered methylation on the IGF-2 gene, the insulin-like growth factor 2, which regulates metabolism and growth. They showed elevated rates of schizophrenia, elevated rates of obesity, accelerated biological aging. Their metabolisms were still running the famine protocol, still hoarding calories, still operating as if scarcity were the baseline condition of existence. 6 decades after the famine ended, and people who had personally never experienced it. Their cells did not know the war was over. Their cells were still eating tulip bulbs. The Irish potato famine, 1845 to 1852. 1 million lives ended. Another million emigrated in conditions barely better than the ones they were fleeing. Trauma so total, so generational, so all-encompassing that it didn't just end people's lives. It broke the social fabric of an entire nation in ways that are still being mapped. Irish and Irish American community studies have shown intergenerational shifts in metabolic pathways, stress response, baselines, and what clinicians kept describing for generations afterward as hereditary melancholia, the great quiet, a sorrow that seemed to exist in the mind, but not in the mind, but in the body. A heaviness that could be traced to any individual specific grief, only to the inherited weight of a catastrophe that had ended before the sufferer was born. Irish literature is saturated with this. Beckett's characters waiting for something that never arrives, O'Brien's people trapped in cycles they cannot escape. The whole tradition of Irish tragic fatalism, which is also, by the way, some of the funniest literature ever produced, because dark humor is also a hereditary survival mechanism, and we will absolutely get back to that. The American Civil War, Andersonville prison camp. If you do not know Andersonville, brief history, it was a Confederate prisoner of war camp in Georgia. 45,000 Union soldiers were imprisoned there between 1864 and 1865. Nearly 13,000 were unalived of disease, starvation, exposure, and violence. It was one of the most lethal environments in American military history. Researchers published a study, long, careful, methodologically rigorous, showing that the sons of the Union soldiers who survived Andersonville had significantly higher mortality rates than the sons of the Union soldiers who had not been in prison there. The sons who were not at Andersonville, who in many cases weren't born until after the war ended, who grew up in peacetime and relative safety. Lives ending younger than they should have were reasons that trace back to something that happened to someone else's body before they existed. And then look at what American Southern Gothic literature has been doing for a century and a half. Faulkner's family is decaying in their ancestral homes, unable to escape the past. Flannery O'Connor's grotesque, haunted characters carrying wounds they didn't earn. The whole tradition of the Southern Gothic is essentially an artistic attempt to make sense of exactly this. The feeling that history had gotten inside the body and cannot be evicted. It's not a metaphor, it never was. It was a description. And here's the thing that doesn't get said enough about folklore as a biological warning system. The cultural taboos that surrounded pregnancy in almost every traditional society, the strict behavioral, dietary, and psychological rules for the expectant mothers, the elaborate rituals for warriors returning from battle, the purification ceremonies, the isolation periods, the specific foods and prayers and practices designed to help a fighter return from a state of war. These were not superstition. They were intuitive epigenetic protection. They were communities that had observed across generations that what a pregnant woman experiences gets into the child, that a warrior who returns home without processing what he went through brings the war back with him, not just in his mind, but in his body, and potentially in his children's bodies. They built elaborate social technologies to down-regulate maternal stress during crucial developmental windows, to prevent the chemical tagging of the unborn child's genome with the signature of trauma. The ritual was the intervention. The story was the delivery mechanism. The community was the therapeutic structure. We dismantled all of that and replaced it with the myth of individual resilience and a 6-week postpartum checkup. I'm not being romantic about the past. The past was also so terrible in many ways, but on this particular point, they were centuries ahead of us. Okay, we have established a pattern. Now we have established the history. We have looked at the case studies. Now I want to get into actual machinery. Because machining is where it gets genuinely mind-bending, and I think you deserve the full picture. How does a nightmare travel from one generation to the next without changing a single letter of the DNA code? It comes down to three interlocking systems, and together they form the most elegant, haunting mechanism I have ever encountered. System 1, DNA methylation. We touched on this in part 1. Let's go deeper. Your genome contains roughly 28 million CPG sites, specific locations in the DNA where the cytosine nucleotide sits next to a guanine nucleotide separated by a phosphate group. These sites are the primary targets of methylation. They are the switches. When trauma triggers the stress hormone cascade, the methyltransferase enzymes that get activated don't go randomly scrawling methyl groups all over the genome. They go to a specific address, the addresses of specific genes, the genes responsible for efficiency. And here is the specific city that should keep you up at night. Different types of trauma leave different methylation signatures. The methylation pattern left by a famine is different from the pattern left by combat. The pattern left by chronic psychological abuse is different from the pattern left by acute physical threat. The epigenome is not just recording that something bad happened, it's recording what kind of bad thing happened. So the descendants can be pre-calibrated for that specific category of danger. It is a biological intelligence system. It is reading the environment, encoding information about that environment, and transmitting that encoded information across generations. It is, by any reasonable definition, a form of biological memory that operates below the level of consciousness and survives the death of it or the death of the organism that formed it. System 2, histone modification. DNA doesn't float loose in the nucleus. It's wound across proteins called histones. Imagine a thread wrapped tightly around a series of spools. The way the DNA is wound determines whether the genes on it can be accessed and read. Tightly wound, the genes are hidden. The cellular machinery can't get to them. They're effectively silenced. Loosely wound, the genes are exposed, wide open, available for constant activation. Trauma can alter the chemical state of these histones through acetylation, methylation of the histones themselves, phosphorylation, changing how tightly they grip the DNA. And these histone configurations, like the methyl tags on the DNA itself, can be transmitted across generations. It is a second layer of the same system, two independent mechanisms for encoding the same information. The body really wanted to make sure this message got through. System 3, the RNA cargo. This is the newest piece of the puzzle and the one that most elegantly explains how the information bypasses the global reset. Inside the sperm, specifically in the cytoplasm and the small RNA payload, there exist molecules called small non-coding RNAs, microRNAs, tRNA-derived small RNAs. These are not part of the DNA sequence. They are not genes. They are regulatory molecules. They control how genes are expressed once the embryo begins developing. When a parent undergoes severe trauma, the profile of these small RNAs and their sperm changes. Specific RNAs increase, others decrease, the payload shifts. Upon fertilization, these RNA molecules are released into the developing embryo, and they do their regulatory work in the earliest stages of development. Before the embryo has even fully implanted into the uterine wall, they alter how the stress response circuitry of the developing brain is constructed. They calibrate the HPA axis, the body's core threat detection system, to a setting that reflects the ancestral environment. Not the environment the child will be born into, the environment that was dangerous enough to warrant encoding this warning in the first place. The nightmare doesn't change the bones of the genetic code. It changes the biological climate in which those bones are grown. And before you ask, yes, maternal transmission works differently, but achieves similar results. When a pregnant woman experiences severe trauma, 3 generations are simultaneously exposed to the chemical cascade, the mother, the fetus developing inside her. And the primordial germ cells inside that fetus, the cells that will eventually become the mother's grandchildren. One event, 3 biological generations, all experiencing the chemical signature of the same catastrophe at the same time. The family curse was never a single moment of bad luck. It was a moment of bad luck that was efficiently, precisely, chemically transmitted to everyone downstream. I need to tell you something that is going to disturb you on a level that is difficult to prepare you for. I'm going to tell you anyway, because I think you deserve the real picture, not the comforting edited version. We have been telling ourselves a story about dilution, about distance, about how, with enough time, with enough generations between us and the original catastrophe, the epigenetic marks wash out. The ghost grows within. By the 3rd generation, the 4th, surely, the inheritance fades. The curse dissolves. The wound closes. A 2025 study in the Proceedings of the National Academy of Sciences would like to set that story on fire. Researchers tracked the lineage of mammals exposed to ancestral toxicant, specifically a compound that permanently disrupted reproductive and stress endocrine pathways across 20 generations, not 3, not 4, 20. They were looking for the fade. Every researcher in this field expected the fade. The methylation marks were supposed to degrade, get overwritten, lose their coherence across the noise of generational variation and combination. They did not fade. The epigenetic scars stabilized across 20 generations. 20. The structural changes in DNA methylation in the descendants match the original ancestral trauma signature with remarkable fidelity. 20 generations removed from the event had no direct exposure to. Descendants were still carrying the molecular fingerprint of something that happened to their great to the 20th power grandparents. That alone is enough. That alone is the most terrifying thing I have ever read in a peer-reviewed journal. But the study didn't stop there, because the real horror wasn't the persistence. The real horror, what happened at the end. By the time the lineage reached generations 15 through 20, entirely new pathologies began appearing, conditions that had not been present in early generations, conditions that had no direct analog in the original trauma. Severe labor complications, reproductive failures, physical manifestations of dysfunction that the researchers had not anticipated and that bore only oblique relationship in the initial event. The ghost didn't fade. The ghost evolved, sitting dormant in the genetic background across centuries, not weakening, not dissolving, but changing, finding new physiological terror to inhabit, developing new expressions of the original disruption in ways that the original environment never produced. A multi-century biological haunting, not diminishing what with time, but mutating, growing new roots. Now let me give you the dark comedy version of this because I think we need it. The idea that your anxieties would naturally dilute away over time, that you would eventually, generationally, just get over it, was always a little optimistic, wasn't it? That's the very human kind of wishful thinking. Surely it can't last forever. Surely time heals all wounds. Tell that to the 20th generation of a mammal lineage that is developing reproductive failure from something that happened to an ancestor. They would need a very, very long spreadsheet to trace back to. Time, it turns out, does not automatically heal epigenetic wounds. Time just gives them more room to get creative. I need to introduce you to a word that is about to change the way you think about yourself as a biological entity. The word is holobiont. A holobiont is not you, or rather, a holobiont is not just you. A holobiont is you plus the trillions of microscopic organisms that have made their home in and on your body for your entire life. Your gut microbiome, the bacteria in your skin, on your skin, the microbial communities in every cavity and crevice of your biology. You are not a single organism. You are an ecosystem, a walking civilization of interspecies cooperation that had been running together since before you were born. You contain more microbial cells than human cells. The genetic content of your microbiome dwarfs the genetic content of your human genome by a factor of roughly 150 to 1. You, the thing that identify as you, the thing that has a name and thoughts and fears and opinions about whether the toast is burnt, you are in a permanent, intimate, deeply independent relationship with a community of organisms that have their own chemical language, their own responses to stress, and their own mechanisms for influencing your nervous system. And when your ancestors was catastrophically traumatized, the catastrophe didn't just write their human cells, didn't just rewire their human cells, it rewired the entire ecosystem. Here's the mechanism. During periods of severe stress, the nervous system floods the gut with stress hormones, catecholamines, adrenaline, noradrenaline, glucocorticoids, the gut lumen. The space inside your intestines where microbiome lives becomes saturated with these chemical signals. The microbes read them. They have specialized surface receptors for these exact hormones. They are tuned to the biochemical language of their host nervous system. They know when the host is in crisis, and they respond accordingly. The beneficial bacteria, the calm anti-inflammatory residents that produce the compounds your nervous system needs to function sanely, they crash. Bifidobacterium, lactobacillus, the organisms responsible for producing short-chain fatty acids, compounds like butyrate and acetate, which acts as a natural inhibitors of inflammatory signaling and critically as natural regulators of histone modification, natural epigenetics stabilizers. When these bacteria crash, the FCFA disappears, and without those molecule guardrails, the genome loses one of its primary natural protection against unchecked epigenetic remodeling, including in the germline, including the sperm and egg. Meanwhile, the opportunistic bacteria, the ones that thrive in chaos and inflammation, explode. They take over the vacated real estate. They set up shop in a gut that is now running a crisis protocol. And they reinforce that crisis protocol with their own chemical outputs. But it gets worse. It always gets worse on this podcast. That's kind of our brand. In cases of severe, prolonged famine, when the gut microbiome has been deprived of dietary fiber that has literally run out of food, specific bacterial strains began consuming the much layer of intestinal wall itself. The productive lining, they eat through it. The gut becomes permeable. Inflammatory cytokines, altered bacterial extracellular vesicles, microbial waste products, they spill into the bloodstream. They migrate through the body's circulatory system. They reach the gonads. They rewire the microenvironments of developing sperm and oocytes. The ancestral microbiome, the traumatized crisis mode, inflammation-saturated microbial community is now actively participating in the epigenetic programming of the next generation. And then, because this podcast because this is a podcast about inherited nightmares, and we are going to see this through to the end. The microbiome is transmitted from the mother to child during birth and early infancy. Directly, vertically, the first microbes a newborn encounters come from the mother's birth canal, from skin contact, from breast milk. From the child is inoculated with the mother's microbial community almost immediately upon entering the world, which means if the mother carries a trauma microbiome, one that has been restructured by ancestral crisis to a state of chronic inflammation and crisis protocol, the child inherits that restructured microbial community. The inherited nightmare isn't only a human cellular memory, it is a full ecosystem panic, multi-species, co-authored by microorganisms that themselves have never experienced anything but the crisis state they were born into. Your great-grandmother's gut bacteria went through something terrible. Their descendants, living in you right now, are still running the crisis response. Your gut microbiome is generally traumatized. I'll give you a moment with that. We have established that you are haunted by your human ancestors. We have established that you are also haunted in a meaningful biological sense by our microbial ancestors. Now I am going to tell you about the oldest haunting of all. It predates humanity by millions of years. Your genome is not entirely human. I want you to sit with that sentence. Your genome, the thing you think of as the blueprint of you, is not entirely yours, not entirely human, not entirely from any species you would recognize. Roughly 8 to 10% of the human genome is composed of endogenous retroviruses, ERVs. These are ancient viral pathogens. They infected our primate ancestors, some of them before the human lineage had fully diverged from other great apes, hijacked their reproductive cells, inserted their genetic sequences into their host genome, and then became permanent passengers. The host couldn't evict them. The immune system couldn't eliminate what had been incorporated into the genome itself. So they stayed, generation after generation, millennium after millennium. They became part of us. Under normal conditions, in a healthy, unstressed organism, the body keeps these ancient viral sequences buried under dense methylation, heavy chromatin packaging. The retroviral genes are fossilized, silent. They are remnants of a biological war that ended so long ago. The battlefield had grown over with new forest. They do nothing. They say nothing. They exist in the genome like an ancient ruin inside a city. Visible if you look for it, but inert. But when extreme transgenerational stress occurs, when the methylation patterns that keep these ancestor sequences silent are disrupted by the same mechanisms we've been discussing, famine, catastrophic trauma, the kind of environmental assault that strips away the epigenetic guardrails, these ancient retrotransposons can wake up. They begin doing what viruses evolve to do. They transcribe themselves. They produce the molecular machinery to copy their sequences, and they insert those copies into new locations in the genome. A phenomenon called retrotransposition, jumping genes, mobile elements, cutting and pasting themselves into the genetic text, altering the instructions in ways that can fundamentally change how the organism's brain processes reality, how the immune system responds, how the nervous system interprets threat. Now, here's the part where this moves from disturbing to generally cosmically unsettling. These are not recent viral sequences. These are not relatives of anything circulating today. Some of these endogenous retroviruses entered the primate lineage during the Eocene epoch. Before our ancestors were recognizably human, they are passengers from an era before language, before culture, before Homo sapiens as a species existed. They have been silent in your genome your entire life. They have been silent across the entire history of your family. They have been silent in the human lineage for millions of years. And the extreme transgenerational stress is one of the documented triggers for their reactivation. You are not just haunted by the memories of your human ancestors. You are haunted by prehistoric microscopic entities that predate your species, that have been dormant in your genome for longer than modern humans have existed, that are waiting in the deepest archive of your biology for the right confluence of ancestral terror to receive their wake-up call. The ghost is not just in the blood. The ghost predates the blood. The ghost was there before your family, before your culture, before your language, before your species had words for what a ghost was. You ask why the fear feels ancient. Because some of it is. Let's talk about the matrilineal curse. Because human cultural intuition landed on this one with startling precision, and it deserves a full examination. In virtually every mythology of the family hex, the maternal line carries specific weight: the mother's curse, the grandmother's shadow, the female bloodline as the source of both deepest blessing and most inescapable doom. Almost no major literary or folkloric tradition of the inherited curse focuses primarily on the paternal line. It's always the mother, the grandmother, the great aunt who life ended in scandalous circumstances and left a shadow on the woman who came after. For a long time, this was chalked up to misogyny. Women as vectors of social anxiety, women as the site of cultural projection. And certainly, misogyny played a role in how these stories were framed. But the biology has an interesting thing to say about this. There's a form of inheritance that is entirely maternal, the mitochondria. You know mitochondria, powerhouse of the cell. You memorized that in school and then promptly never thought about it again. But here's what the textbook didn't linger on. Mitochondria have their own DNA, separate from the nuclear genome, a small circular independent genetic code that runs the cell's energy production. And mitochondria are inherited entirely from the mother. At fertilization, the embryo actively and deliberately destroys the paternal mitochondria. The sperm carries its own mitochondria. It needs them to power the swim. But the moment fertilization occurs, the embryo triggers a targeted degradation pathway that eliminates the paternal mitochondria payload. Every time, without exception. The child inherits nuclear DNA from both parents, but the mitochondria, every single one, comes exclusively from the mother, which means when a maternal ancestor experienced severe, prolonged, chronic trauma, the kind of sustained psychological crisis that generates a massive oxidative stress, the oxidative damage accumulated in mitochondria, in their membranes, in their DNA, in the enzyme systems that govern energy production. And she passed 100% of those damage, reconfigured, crisis-adapted cellular engines to her children, who passed 100% to their children, with no paternal contribution to dilute or counteract the pattern. The mitochondria in your cells right now are the direct, uninterrupted descendants of your maternal ancestors' mitochondria. If her mitochondria were structurally altered by trauma, calibrated for a state of chronic crisis, tuned to operate in an environment of threat, you are running those same engines. An immune system that runs chronically inflammatory, a metabolism that swings unpredictably, energy systems that excel under acute stress and collapse in sustained safety, because they were engineered for a crisis that had long since ended. Your grandmother's engine, running in your century. And now, and the cruel irony, the specifically dark humor of the mitochondrial curse is that the engine designed for wars runs catastrophically in peacetime. The adaptations that kept your maternal ancestor's body functional in a life or death crisis are precisely the adaptations that make life in a stable, relatively safe environment feels somehow wrong, itchy, like wearing armor to a dinner party. You feel the most alive in chaos. You feel strange in stillness. You have more energy in a crisis than most people have at baseline. You find peace suspicious. You are not built for this world. You are built for hers. And nobody told you that the building was inherited. We have spent the better part of this episode establishing a scientific fact. Targeted specific trauma can encode heritable biological changes precisely, predictably, with outcomes that persist for generations and that physically restructure the sensory and stress response architecture of descendants. I want you to hold that fact in your mind for a moment, and then I want you to follow me down a very specific rabbit hole, because there is a theory. It lives in the fringe and the overlap between defense technology circles and molecular biology, where the people who worry about things that haven't been confirmed and the people who know things that can't be disclosed occasionally occupy the same uncomfortable intellectual space. And a theory runs like this. What if someone figured out how to do this on purpose? It has a name. Informally, mnemonic weaponization. The engineering of heritable trauma, not brainwashing an individual, not manipulating a population's beliefs. Something far more ambitious, far more patient, and far more permanent. The allegation, by subjecting a captured or targeted population to highly controlled, chemically amplified trauma, the right sensory input delivered with the right pharmacological enhancement at the right developmental window, you could theoretically encode specific phobias, behavior convulsions, or neurological vulnerabilities into the gremlin of that population, not their minds, their bloodlines. You would not be creating a generation of traumatized people. You would be creating a dynasty of descendants who carry a heritable biological handicap they did not choose, cannot identify, and cannot trace any events in their own lives that experienced it. The targets would have no idea they had been targeted. Their children would have no idea. Their grandchildren would be experiencing anxiety and dysfunction that feels entirely internal, entirely personal, entirely their fault, while carrying the engineered molecular signature of someone else's deliberate agenda. Is there confirmed evidence this has been done before? No, there is not. Not to my knowledge, a declassified program with this specific objective. But let me tell you what is confirmed. The mechanism works. The DS Wrestler cherry blossom experiments proved that a specific sensory input paired with a specific adversive stimulus produces a specific heritable fear response that physically restructures descendant neuroanatomy. The pharmacological application of stress responses is a well-documented area of DARPA and DRPA adjacent research. Compounds that enhance memory consolidation, including the traumatic memory consolidation, have been studied extensively. The military intelligence complex has historically been very interested in behavioral modification, as documented in projects that has been since declassified, and which I will not name specifically because this is a podcast and not a legal brief, but you know the ones. The science to do this has existed in component parts for decades. What would be required to weaponize it primarily? Ethical vacancy, sufficient resources, and patience measured in generations. History contains no shortage of institutions with all three. I am not telling you this happened. I am telling you that the mechanism exists, that interested parties exist, and that the design of such a program would produce exactly the kind of evidence that is most difficult to trace. Not a smoking gun, but a biological signal in a population that looks from the outside like ordinary generational anxiety. Draw your own conclusions. That's what you're here for. And while you're at it, consider the secondary conspiracy angle, which I find equally fascinating and significantly more verifiable. John Baptiste Lamarck was the biologist who, in the early 19th century, proposed that organisms could pass on traits acquired during their lifetime. The classic example, a giraffe stretches its neck reaching for leaves and passes a slightly longer neck to his offspring. Darwin's framework of genetic mutation and natural selection buried Lamarck so thoroughly that his name became scientific shorthand for discredited thinking. Lamarckian inheritance was the polite way of saying wrong about everything, except epigenetic inheritance is, in its fundamental mechanism, Lamarckian. An organism acquires characteristics during its lifetime, through experience, through environment, through trauma, and passes those characteristics to its descendants, not through genetic mutation, but through the epigenetic layer, which means that for roughly 150 years, the scientific establishment dismissed, mocked, and actively suppressed research that validated the core of Lamarck's intuition. Some fringe theorists go further. They allege that early epigenetic research was specifically deliberately buried, not just through institutional inertia or the natural slowness of paradigm shifts, but through active suppression, because validation of Lamarckian inheritance threatens several very powerful interests simultaneously. It threatened the DNA as destiny model that underpins the genetic testing industry. It threatens the pharmaceutical industry's model of treating inherited mental illness as a fixed neurological deficit rather than the environmentally induced, potentially reversible epigenetic condition. And most dangerously, it created a biological basis for holding corporations, governments, and institutions legally liable for the multi-generational biological damage caused by environmental catastrophes, systemic violence, and economic exploitation. If you can demonstrate that a corporation's decision to dump toxic chemicals in a community created heritable epigenetic trauma that affected that community's descendants for 20 generations, you are no longer talking about an environmental lawsuit. You are talking about something that sounds uncomfortably close to a crime against a lineage. I cannot verify the active suppression claim. What I can verify is the epigenetics that is arriving late to the scientific mainstream, despite evidence that had been accumulating since the mid-20th century, and that the industries most threatened by implications are also the industries most influential in shaping research funding priorities. Make of that what you will. I'm just a Phantom Navigator. I just drive the boat. Here's where I'm going to do something unusual for the paranormal podcast. I'm going to give you hope, not the soft, vague, inspirational kind, the specific, technical, this is actually happening kind. Because 2025 and 2026 have delivered tools that when you understand what they're for, read like science fiction, but they are not fiction. Your epigenome contains approximately 28 million CPG sites, 28 million individual locations where methylation can occur and where the story of your ancestry is written in chemical notation. Mapping this manually was never possible, even with modern sequencing technology. The sheer scale of methylome meant that identifying which marks corresponded to which ancestral events in which populations across which generations was an analytical problem too large for human processing. Until diffuse-based AI models arrived, these systems train on vast multi-generational epigenetic data sets from the diverse populations and catastrophes can ingest a patient's blood or saliva sample, sequence the methylation patterns across the full epigenome, and identify what researchers now call trauma fingerprints, distinct epigenetic signatures that correspond to specific categories of ancestral events. And utero famine looks different from a combat trauma, looks different from chronic maternal anxiety, looks different from acute environmental toxicant exposure. The AI can distinguish them. It can locate them in methylome with a precision that no human researcher could manually replicate. And then, because the AI models are also trained on three-dimensional chromatin architecture, they can trace how specific methylation mark at a specific CPG site alters the way the DNA folds in three-dimensional space inside the nucleus of a neuron, and map how that altered folding changes the expression of stress response circuits, the amygdala, the prefrontal cortex, the hippocampus. You can give a researcher your saliva. They can feed the methylation data into an AI pipeline, and they can render visually a three-dimensional map of the altered neural circuitry, that chemical mark left by a catastrophe that occurred to someone else before you existed has produced in your brain. We can see the ghost architecture. We can draw its floor plan. CRISPR off. Now let's talk about what happens when you found the address. Traditional CRISPR is scissors. It cuts the DNA strand to physically remove or alter a sequence, powerful but permanent, and imprecise enough in its current iterations to cause off-target cuts, mutations in locations you didn't intend to touch, that raise legitimate safety concerns for therapeutic use. CRISPR off throws away the scissors. It uses a catalytically dead Cas9 protein. The dead here is precise. This Cas9 has been specifically engineered to retain its navigation capabilities, its ability to find a specific address in the 3 billion letter genome with extraordinary precision. While having its cutting function removed, it can travel, it can locate, it cannot cut. When it arrives at the target, say the promoter region of FKBP5, the stress regulator has been jammed in permanent overdrive by 3 generations of ancestral trauma. It binds, and then a molecule domain fuses the dead Cas9 called the CRAP CRAB repressor recruits a set of host enzymes. These enzymes strip acetyl groups from the histones around the target gene and deposit methyl groups in their place, forcing the local DNA to fold into a dense, tightly wound configuration called heterochromatin. The gene is not muted. The gene is muted, not deleted, not destroyed, not permanently altered at the sequence level. Muted, as if someone found the volume dial on your inherited alarm system and, gently but firmly, turned it all the way down. The sequence is intact. The door is still there if you need it, but the thing that has been screaming through it for a century can no longer be heard. In 2026, the delivery mechanism for CRISPR-off has been substantially optimized using lipid nanoparticles, tiny fat-based capsules that can carry the engineered mRNA into targeted cells without triggering immune response. The technical barriers that made this therapeutic fantasy a decade ago are not gone, but genuinely reduced. The AI finds the ghost address, the CRISPR-off knocks on the door, and installs a very effective soundproofing system. Now, for those of us who are not quite at the point of epigenetic gene silencing therapy, which most of us, let's be honest, there is a third technology that operates at the somatic level in real time and is already in clinical use. Vagus nerve stimulation, specifically transcutaneous auricular VNS, VNS, which stimulates branches of vagus nerve through the skin of the ear non-invasively without surgery. The vagus nerve is the longest nerve in your body. It runs from your brainstem down through your neck, through your chest, your abdomen, and the primary highway of the parasympathetic nervous system, the system that tells your body it is safe, that it can rest, that the threat has passed, that the alarm can stand down. When the vagus nerve is functioning well, it acts as a natural counter to the sympathetic fight or flight response. The two systems are supposed to be in balance. Threat appears, sympathetic fires, body mobilizes, threat resolves, vagus nerves activates, body recovers. In people carrying inherited epigenetic trauma, people whose amygdala had been pre-calibrated to treat the baseline world as a threat environment. The parasympathetic system is persistently outgunned. The alarm is louder than the all-clear sign. The body never quite fully recovers. It lives in a state of chronic low-level mobilization. Transcutaneous ocular VNS sends calibrated electrical impulses through the skin of the ear to the vagus nerve branches. Those pulses travel to the nucleus tractus solarius, a brainstem structure that serves as a relay station for the parasympathetic system. The effect cascades. Amygdala hyperactivation decreases. Inflammatory cytokine production drops. The nervous system's threat detected baseline is, over the time and repeated sessions, recalibrated. And here is the downstream epigenetic effect that makes this more than symptom management. When a body is consistently held within its window of tolerance, when the chronic cortisol and catecholamine blood finally subsides, the cellular microenvironment changes. The gut microbiome, no longer bathed in stress hormones, can begin to recover. The FCFA producing bacteria can reestablish, and those SCFAs, butyrate, acetate, return to their role as natural epigenetic stabilizers. Over time, in a body that is no longer running a chronic crisis, the volatile methyl tags, the ones that were laid down reactively in panic in response to a threat that no longer exists, begin to be naturally processed out. The ancestral alarm system, deprived of the chemical fuel that sustained it, begins to quiet. You cannot heal overnight. You cannot reprogram 20 generations of epigenetic inheritance with one session in a therapy chair, but the mechanisms are real. The direction is real. The biological capacity for the nervous system to shift given right environment and the right tools is real. And then there's spermidine. I promised you the supplement angle, and here I am, a person who keeps their promises. Spermidine is a natural polyamine compound found in your cells and in a range of foods, aged cheese, mushrooms, soy products, natto. It has been studied for years as a longevity compound, primarily for its role in activating autophagy, the cellular maintenance process by which cells identify and remove damaged components. In 2025, a landmark study confirmed something more specific. Spermidine activates autophagy through a distinct mechanism involving hypoxenation, a specialized protein modification process critical for efficient cellular energy management. And critically, it was shown to preserve mitochondrial function and cognitive performance in aging biological models, even when other protective measures weren't in place. For our purposes, this is really interesting because it's just a pathway, imperfect, partial, but real, for addressing the maternally inherited mitochondrial damage we discussed. The cellular engines that were restructured by your grandmother's chronic stress, the mitochondria running hot in a world that doesn't require it. Clinical trials in 2025 and 2026 are mapping how spermidine, combined with anti-inflammatory protocols, can dampen the specific biological aging acceleration. The epigenetic clock running too fast, that famine and severe stress events leave in descendants. Not a cure, not a reversal, but a form of maintenance, a way of servicing your grandmother's engine so it doesn't burn through you before it's time. We are nearly at the end. I want to use this last section carefully because I think it matters. We have spent a lot of time in the dark tonight. That was intentional. The dark is where the real information lives. the comparable summit version of this topic. Ancestral trauma is real. Healing is possible. You are more than your history. It's true, but it's incomplete. It glosses over the actual mechanism, the actual scale, the actual persistence of the thing. We do not gloss over things on this podcast, but I also don't want to leave you wrecked in a ditch. So here is what the fully picture actually offers. When you look at it honestly, the epigenetic system is not a prison. It is a communication system, one that runs in both directions. If a gene can be silenced by trauma, it can be unsilenced by safety. If a methyl tag can be placed by catastrophe, it can be removed slowly and perfectly over time by an environment that consistently tells the nervous system that the war is over. Cognitive behavioral therapy has been shown to physically alter DNA methylation profiles, not as a metaphor, as a measurable, documented change in the epigenome. The release of oxytocin during general social connection, the production of BDNF during meaningful movements and learning, the reduction of chronic cortisol through consistent safety. These are not just psychological events, they are molecular events. They act as biological erasers, gradually, patiently removing the archaic methyl tags that kept the alarm locked on. EMDR, eye movement desensitization, and reprocessing produces changes in how traumatic memories are encoded and accessed that correspond to measurable neurological restructuring. Somatic experiencing works with the body's own completion cycles to discharge stored threat responses that the nervous system never finished processing. These are not feel-good interventions. They are, in the language we've been speaking tonight, epigenetic editing tools, slower than CRISPR-off, less precise than AI methylome mapping, but available now, accessible now, operating through the same biological system we've spent this whole episode discussing. Healing is not metaphorical, it's molecular. And here is the reframe I want to offer you, the one that has stayed with me the most throughout this research. Every person in your lineage who passed on a heritable trauma response survived the thing that created it. Every single one. Every ancestor whose chemical you are carrying today, but with a person who faced something unsurvivable and survived it, who faced a famine and found something to eat, who faced a war and made it home, who faced a persecution and kept moving, who faced the loss of everything and somehow continued to exist long enough to pass their biology forward. The hypervigilance you carry is not malfunction, it is armor. It is an early warning system engineered by the most motivated possible designers. People who desperately need their bloodline to detect danger faster than organisms without that warning. It worked. The proof is that you exist. The problem, the specific exquisite tragedy of this whole thing is that the armor is calibrated for a world that is gone. The alarm that is tuned to a frequency that the modern environment rarely broadcasts, and so it fires and fires and fires in response to shadows and silence and the ordinary uncertainties of a life that is, by historical standards, extraordinarily safe. You are wearing your great-grandmother's armor in a world she never got to see. She gave you the best tools she had. She didn't know you needed a different one. The task And I say this as gently as I can while still saying it clearly. It is not to be ashamed of the armor. The task is not pathologize. Is not to pathologize it or medicate it into silence. This or spend the rest of your life fighting it. The task is to learn to read it as what it is, a message from someone who loves you across time, who wanted you to survive, who did everything a body can do to make sure the warning reached you, and then gently, carefully, with whatever combination of therapy and science and community and time is available to you, to write back. The war ended. I'm okay. You can rest now. That message, when you can generally send it, not as a thought, but as a felt somatic cellular experience of actual safety, travels the same pathways in reverse. It doesn't undo the past. Nothing undoes the past. But it begins the long, slow, real process of recalibration. You are not the curse. You are the latest chapter of the longest survival story ever told. You have the scientific vocabulary now to understand what you're carrying. You have, for the first time in human history, tools that can read the molecular address of an inherited fear and begin, carefully, imperfectly, promisingly, to respond to it. Your ancestors survived. You are here. The ghost is real. The science is real. And the possibility of laying it down. And so is the possibility of laying it down. That's the bloodline curse, decoded. Not in metaphor, not in mythology. Through the mythology, as it turns out, was never wrong about basic facts, only about the mechanism. In chemistry and molecular biology and language of methyl groups and stress hormones and microRNAs that slid past the embryonic reset like contraband across a border that was supposed to keep them out. The ghost is real. It's written in the most intimate possible language, the chemical architecture of your actual cells. It's in the way of your stress response fires, the way your gut reacts, the specific, personal, precise shape of fear that visit you when you haven't slept enough and the lights are off and there's nothing externally wrong. That's your great-grandmother's handwriting. That's the oldest letter you've ever received, written in a language you weren't taught to read, but your body has known how to respond to since before you were born. And here's what I want to leave you with tonight. You have been walking around thinking the fear is yours, thinking it says something about you specifically, your weakness, your irrationality, your inability to just cope better, thinking it's a flaw in the manufacturing. It is not a flaw, it is a message. The ancient chemically encoded, biologically transmitted message from someone who needed you to know. The world can turn on you. Here's what it felt like. Here's the alarm I built so you know if it came back. The tragedy isn't the message. The tragedy is the body handed you a translation guide. The tragedy is that nobody handed you a translation guide. You have one now, so take it, use it. Sit with the extraordinary, unsettling, strangely comforting reality that your inexplicable anxieties are not the sign of a broken mind. They are evidence of an unbroken lineage of survival, repeated, of people who made it through the unsurvivable and found a way to reach forward in time and warn you. The alarm is miscalibrated for the world you live in, but it was built with love, the only white language available. I'm your host, This has been the Phantom Navigator. I'll see you next time. Somewhere between the data and the dark, stay curious, stay haunted, and whatever you do, don't ignore the fear you can't explain if it knows something you haven't remembered yet.